The synapse. We've talked about the synapse before. Before we talk about the synapse, let's establish some new terminology for the synapse. So we know that a synapse is a space between two cells, right? And that little tiny space right there, this would be a enhance of that little tiny space. We know that the feet or the toes of an axon of a neuron are called the teleodendria, and the toes are called the axon terminals. And you guys know that the neurons, they don't actually touch, but there's a space, a very small space that separates them called the synapse. So it can be a space between a neuron and a neuron, or it can be a space between a neuron and a muscle fiber. Either one. When it's a space between a neuron and a muscle fiber, we call that the neuromuscular junction. We've already talked about that. We're now going to talk about the synapse as though it were a space between a neuron and a neuron. In order to do that, let's establish some terminology. The neuron on the left is going to be called the presynaptic neuron. All that means is that it's the sending neuron. It's the neuron that will release its neurotransmitters into the synapse. It doesn't always have to be on the left, just saying. In our book, though, and everything we draw, it'll pretty much be on the left. The receiving neuron is called the postsynaptic neuron. In order to help you with this, to make it abbreviations, you can just do presyn or postsyn. And I will also use those abbreviations as well. Okay, so this is the presynaptic neuron. This is the postsynaptic neuron. This space in here is the synapse. So obviously this picture is like totally enhanced. I made this picture a long time ago. I think in like 2010 when I first started teaching at FLCC. Yeah, They don't actually all go to the same place. Neurotransmitters can end up on multiple areas. There are channels that receive them in all places. They all don't go to one place. I've never fixed it. There are two kinds of synapses, and we will only be considering one kind of them here. The other kind is considered when we do cardiac muscle next semester. The first type, and I just want to go over it quickly and then we'll set it aside, these are called electrical synapses. So there is a space that separates the cells. However, there are channels between them. And actually this is a muscle fiber. It's a cardiac muscle fiber. And so is this one. So this is how muscle fibers communicate in terms of synapses. This isn't really, well, we'll talk about that. Just set that aside. The idea, though, is that they kind of share their cytoplasm with these channels. So, I mean, it's almost like the heart's one big cell. Since they all share cytoplasm, are they really separate? Anyway, we will be considering chemical synapses. These are synapses that use neurotransmitters. There are no neurotransmitters on the picture on the right. There are neurotransmitters in the picture on the left, right? We have some up here tied up in vesicles. We have some being puked out into the synapse here. And then we have some connecting with receptors. So these are puked out in the synapse, which is a full-on gap between the cells. There's nothing spanning that gap. These are chemical synapses, and this is what we will be considering. There are tons of neurotransmitters. There's lots of them. We need to remember that we talked about acetylcholine. Acetylcholine helps neurons communicate to muscles at the neuromuscular junction, but acetylcholine is also used in your brain. There are 3 other neurotransmitters I want you guys to get to know, actually 4. There's norepinephrine, sometimes called noradrenaline. That's not the correct terminology. Call it norepinephrine. We're going to abbreviate that as NE. Yeah, you could associate it with adrenaline. Yes. In fact, epinephrine is also a neurotransmitter. We call that E. But norepinephrine does the same things that adrenaline does. It keeps you awake and alert. It's an excitatory neurotransmitter. There's also a neurotransmitter called dopamine. Dopamine is more involved in your pleasure pathways, like in your hypothalamus, thirst, hunger, sex, addictive behaviors. Dopamine can be inhibitory. It can be. In fact, in your brain, it can regulate precise movements. So raise your hand. And you raised your hand very precisely. It didn't like knock over to the other side. That's because when you were done raising your hand, dopamine inhibited that signal from your brain. People with Parkinson's have a lack of dopamine, which is why they have jerky movements. Dopamine can also be excitatory, and this is where it acts in what is called your limbic system. Your limbic system is your emotional seat, and any addiction is a product of your limbic system, and addictions can fiddle with dopamine. I mean, directly, morphine basically mimics dopamine, and you give your body tons of dopamine, and then you take it away, and you go through withdrawal. The other one I want you guys to know is serotonin, which also acts in addictive behaviors, but it's more intense in terms of addictive behaviors. Serotonin's about obsessions and compulsions. You may know that serotonin is the happy neurotransmitter. Yeah, I'll give you that, but serotonin is a neurotransmitter that can help you regulate with small changes in homeostasis instead of big changes in mood swings. So it's more of a regulator than a make you happier. There are some types of chemical synapses. Your book talks about these in chapter 12 and 13, kind of like you know what they are. So let's just establish some terminology. We'll use this a little bit later when we talk more specifically about the brain. But there are cholinergic synapses in your body. We've already talked about those, right? These are cholinergic, acetylcholine. These are synapses that release acetylcholine. The neuromuscular junction is an example of that. Cholinergic synapses are always excitatory. Think about it. The neuron excites the muscle fiber. So whether these types of synapses are in your brain or in your body, they're always excitatory. They always use ACH. You have a great example of this, that any neuromuscular junction is a cholinergic synapse. The other synapses are called adrenergic, and you might think, oh, those are the ones that give me an adrenaline rush. No, they're related to that. But their word or their naming, adrenergic, comes from the fact that they release epinephrine and norepinephrine. And these are, they can be excitatory or they can be inhibitory. I want to draw you guys a picture of a synapse right now. So this is our, let's label all of this. This is our presynaptic neuron. And let's do this. This is our postsynaptic neuron. You're like, Amy, why didn't you draw the wavy folds? Because it's not a muscle fiber. Any neuron might have a vesicle with acetylcholine in it. The neuron might have a vesicle with epinephrine in it. Or the neuron might have a vesicle with norepinephrine. Honestly, it probably has all three and more that we didn't talk about. On the other side, there are tons of protein receptors, right? Protein receptor, protein receptor, protein receptor. And some of them receive ACH, and some of them receive epinephrine, and some of them receive norepinephrine, and some of them receive... So you see how it's not so simple. We're going to draw synapses and explain them as though there's one neurotransmitter being received and transmitted. That's not the case. The case is that there are kind of all of them in all of your neurons. I like this diagram because we've seen it before, right? We've seen this picture on the top here. In fact, we could label the presynaptic neuron. We could label this as the postsynaptic neuron. We can label the synapse. This is the axon terminal, right? These little guys here are protein receptors. And we know that the neurotransmitters are going to connect with these protein receptors, but not move through them. Thou shalt not pass, right? Remember, it's sodium that moves through and potassium that moves through them. What is this little picture over here that I have on the right? This is kind of new to us. This little picture over here is showing you how a neurotransmitter can get removed from a synapse. And you already know one of these ways. We talked about ACH when we talked about muscle fibers, and ACHE, which was the enzyme that degraded ACH. In fact, all of your neurotransmitters have their own enzyme, and therefore all of them can be degraded by an enzyme. That's the first way that they can be removed from a synapse. The second one is that they just walk away. Neurotransmitters are like, yeah, I'm done with you. No, neurotransmitters are in concentration in the synapse, so they move away from that area. They're concentrated there. There's too many of them. The other way is that your neurotransmitters can get sucked back up by the presynaptic neuron, right? It puked them out and it can suck them back up, kind of like watching a video in reverse of someone puking. In order to do this, they don't just move through the membrane. There has to be a specific protein that helps them do that. And sometimes these proteins, which are called reuptake receptors, sometimes they get confused and they start puking out the receptor instead of taking it up. Sometimes they get blocked. In fact, there are tons of drugs called SSRIs. And what they do is they inhibit the reuptake of serotonin. We'll talk about them. Just hold that thought. Many antidepressant drugs work at the synapses in your central nervous system, in your brain, your limbic system that controls emotional input and output. But one type of drugs that is widely used are called SSRIs, and those are selective serotonin reuptake inhibitors. So these drugs, they prevent serotonin from getting sucked back into the neuron, and it keeps serotonin in the synapse. And by keeping it in the synapse, the postsynaptic neuron can continue to have action potentials. So these kinds of drugs are really commonly known. Paxil, Prozac, Zoloft, Wellbutrin, Lexapro, Cymbalta, a whole bunch of them. And they specifically work on serotonin. They're selective, and they prevent this process right here. What they do is there's a molecule of the SSRI that sits on the plasma membrane of the presynaptic neuron, and it prevents that neuron from sucking back up the serotonin. Okay, all right, we can talk about the steps of an action potential. And actually, we've already done that. We've used this picture on the left, but your book likes to use this picture on the right. So before you even, look at anything, you got to kind of like label the anatomy. We seem to have mitochondria up here. This up here apparently is the presynaptic neuron. This one down here would apparently be the postsynaptic neuron. These are our lovely protein receptors, right? And we also know that these are also channels. They'll open up to let things through. So, these are chemically gated channels. This channel up here is a voltage gated channel. And we now know that this channel lets calcium in. It's specific to calcium. Over here, we have some vesicles, and these vesicles have ACH in them. We're really used to ACH. These vesicles could have serotonin, dopamine, or a whole host of other neurotransmitters that are in the brain. We'll use acetylcholine because no matter where it's used, it is always excitatory. Therefore, its purpose is to always spark an action potential in the postsynaptic neuron. Oh, look, I have labels on this myself. Sorry. Oh, even more. The first thing that has to happen at a synapse is that an action potential has to arrive at the axon terminal of the presynaptic neuron. And this is what your book is indicating here by this red arrow, both of these red arrows. I like to draw action potentials like this because they really indicate the movement of sodium and potassium in and out of the cell membrane. This slide has a few steps on it. Okay, so we can see the little red piece right here indicating that we have a change in voltage. And that's what opens this voltage-gated calcium channel. So the membrane changed in its potential and this voltage-gated channel opened up. And the only thing that moves through this voltage-gated channel is calcium. No other ion molecule can fit through it. What calcium does once it enters into the neuron, right? I mean, it was in the outside of the neuron. Now it's in the neuron. And what it does is it triggers the downward movement of these vesicles, and they puke out all of their acetylcholine into the synapse. And we're familiar with this process, right, of exocytosis of the neurotransmitter. And again, just remember, this could be any neurotransmitter here. We're talking about the brain. We could use dopamine, other things like that. So think about this. You could take a drug that prevents this process with some kind of neurotransmitter. There aren't many drugs that do that, though. There just aren't. We haven't been able to fiddle so much with this process. Once our neurotransmitter is in the synapse, it can then bind to those protein receptors. And when it binds to those protein receptors, they're chemically gated channels, not voltage gated channels. And when these channels bind to their neurotransmitter, the channels will open, allowing sodium to enter into the postsynaptic neuron. Let's note that here. It enters into the postsynaptic neuron. And this causes depolarization, which is the first step to having an action potential. And you can see down here that this diagram indicates an action potential in the postsynaptic neuron. So this process is about stimulating the postsynaptic neuron. And of course, in order to stimulate the postsynaptic neuron, you need to have neurotransmitters in the synapse. I just want you guys to note that the neurotransmitter does not enter the postsynaptic neuron, right? It's like the token that gets put in the protein receptor so it opens up. So the idea with many antidepressant drugs, which are also used for pain mediation, is that they do fiddle with areas at the synapse, and they try to keep certain neurotransmitters in the synapse. That's the goal of many of our drugs. So, I mean, there are drugs that serve to remove it from the synapse, but most of our antidepressants and pain medications, they serve to keep neurotransmitters in the synapse. This picture here shows you a presynaptic neuron that could be stimulating an action potential in the postsynaptic neuron. And of course, we can have drugs that affect the processes in either of these neurons, but we've found that there are other neurons that get in on the party. And what they do is they can alter the rate of the neurotransmitter release. They can influence the presynaptic neuron to puke out all of its neurotransmitters or some of its neurotransmitters. And the neurons that do that are called neuromodulatory axons. Some of that, so these guys over here are changing the rate at which the presynaptic neuron releases. This one over here is changing the rate at which the postsynaptic receives the neurotransmitter. So this neuromodulatory axon up here is maybe inhibiting or exciting the postsynaptic cells. So it's like, oh, ooh, ooh. Drugs that do this, most of the drugs that do this are called opioids. They work on neuromodulatory axons to influence synapses to either keep neurotransmitters in the synapse or remove neurotransmitters from the synapse. The thing that you guys that you have to remember is that it's not just about keeping the neurotransmitter in the synapse. If it's in the synapse, it will excite the postsynaptic cell. That's the thing. If it's not in the synapse, we don't get a transmission of action potential. So it's not just about having neurotransmitters in the synapse. They have to be able to do their job and excite an action potential in the postsynaptic neuron. This diagram in your book can be a little bit complicated, but what we're talking about here is we're talking about the yellow neuron trying to hook up with the purple neuron, and the green neuron gets in the way. And this is what is called presynaptic inhibition. If we look at this more intense diagram on the bottom, the yellow is our presynaptic neuron. The purple is our postsynaptic neuron. And the yellow wants to spew out all of its neurotransmitters into the synapse so it can excite the postsynaptic neuron. But what's happening is that the green is releasing its own neurotransmitter that then inactivates the calcium channels. No calcium moves in, and there's no exocytosis of the neurotransmitter. Think about this again. Yellow is trying to communicate with purple by releasing its neurotransmitters, but green is releasing its own neurotransmitter that blocks these calcium channels. This is what is called presynaptic inhibition. This neurotransmitter here, GABA, it is a neurotransmitter in your brain, and yes, it inhibits calcium channels. Certain drugs can increase GABA in your brain. And what it does basically is it stops action potentials from getting from one place to another. So GABA is like an inhibitory neurotransmitter, which alcohol influences. The other way that we can do this, obviously, is we can increase the yellow neuron's ability to hook up with the purple neuron. So what we want to do here is facilitate. We want to encourage the connection between yellow and purple. We want to make it so that yellow spews out more of its neurotransmitters into the synapse and thus can stimulate purple to have an action potential. So green, what green does in this situation is it releases serotonin. And serotonin does just the opposite of GABA. Serotonin makes it so these calcium channels are more likely to allow calcium in, or they allow more calcium in, which makes more exocytosis. And again, we're exciting the postsynaptic neuron more and more. So we have presynaptic inhibition and presynaptic facilitation. Actually, ecstasy does just this. It increases the activated calcium channels, making the postsynaptic neuron have action potentials when there is actually no sending action potential. And that's what a hallucination is. There's no sensory input, but there's like motor output, you could say.